students, I hope all of you are doing well. I am Honey Rachel and I teach biotechnology in K.B.N college. In this new session, we will try to understand a new concept. What's it? So before I start, I want you to look at this picture. Yes, you see some molecule and its name is insulin, and you see the names of few other molecules which are present in our body. And every molecule that you see, the name that you see here, plays a very crucial role in our body. Consider insulin. It helps in absorption of energy that has been digested in the stomach. And consider amylase. It's a wonderful enzyme that digests the carbohydrates. Consider collagen. It's a structural proteins which make up our body. Consider hemoglobin. It's a wonderful molecule which is present in our red blood cells, and it carries oxygen to all parts of the body. Every molecule that you see here has different unique function, right? And few are hormones like insulin. Insulin is a hormone. Few are structural proteins like hemoglobin, keratin, collagen, histone, and few are enzymes, amylase and protease. Yes, they have several different functions, but you see one common thing in all of these. Guess what? Yes, you are right. The common thing in all of these is these are proteins. All of these are proteins. Biologically and structurally, the basis of formation is these are proteins. They all are made up of simple amino acids which are linked together by polypeptide bonds. And when they come together, when that polypeptide chain come together and form a structure, that protein would look like this. This is basically a structure of insulin protein. So the question is, how are these proteins synthesized in our body? Today let us try to understand the concept how all these proteins are synthesized in our body. Okay, so for that, who explains this concept is, yes, central dogma of molecular biology. This theory tries to explain how a protein is made. So look at the flow. This theory says that from the genetic material, that is DNA, from that flows the information from DNA to RNA, and from RNA our body understands the code and synthesizes a new protein. So central dogma says the genetic information flows from DNA to RNA and then to protein, and that's how every protein molecule is made. Shall we understand this central dogma theory in detail? Yes. So here you see is the DNA. Yes, the DNA, when it is condensed, when it is coiled and it is constricted, it looks like this. Yes, like this. This is chromosome. In cell, there is little space, right? And in that little space, if a long DNA has to fit, it has to be condensed. It has to be in a condensed form. So in cell, basically, and in many times, the DNA exists in the form of chromosome. And in chromosome, we have DNA as genetic material, and in DNA, we have many genes. Yes, many genes. What does a gene mean? A gene means a particular code which codes for a particular protein. Yes. Consider this yellow region. The first yellow region is a gene for insulin, which means here if the gene decodes, then comes the protein insulin, And here we have another gene, amylase coding gene. And here we have hemoglobin gene, keratin gene, actin gene. All these are proteins. And the information which is necessary for the proteins is coded in the DNA. That's why you know what we call DNA. We call it a blueprint. Yes. For a construction of a building, we need a blueprint, right? Even for construction of any protein, we need the blueprint, and that blueprint is DNA. This DNA is present in every cell of the body, right? So as we have the code in the DNA, we want that code to be decoded and we want the protein. Yes. Whenever the body needs certain protein, for example, you have had some food and it is digested, and now you want that energy to be absorbed into every part of the body, every cell of your body. Now there is a need of a protein called insulin, because only insulin can push that energy into the cells. Without insulin, energy will stay in the bloodstream without being absorbed. Yes. Now there is a need of insulin. Yes. Whenever there is need of a particular protein, what happens is a gene is activated. Yes. Here we have a gene. A gene is activated whenever the body needs protein. And what happens? The gene is decoded and the protein is made. Yes. It happens in several steps. Which steps? Let us understand. First, a gene is activated. For example, let us take an example, insulin. Yes, we need insulin protein. For that, first, a gene has to be activated, and then from the gene, a very similar copy of that gene is made, but it's not in the form of DNA. The copy will be in the form of RNA. Why? The DNA, which is the blueprint, the code, whenever it has to be converted into protein, cannot be cut and traveled out for the protein synthesis. The blueprint, if destroyed, there won't be further existence of the cell or protein. That's why our body made it a wonderful concept of making a copy of RNA so that the blueprint exists like that forever until the cell dies. Yes. What happens in the first step? The DNA, it is made into a copy of RNA. And for that we need some machinery. Basically, the head of this machinery is a polymerase, RNA polymerase. Yes, in the first step, DNA is decoded or copied into a single strand RNA. We call it mRNA. We call that process transcription, the first step of central dogma. After the RNA is ready, RNA travels out from the cell or from the nucleus into the cytoplasm, and there it finds a ribosome. Ribosome is an essential molecule which helps in decoding the RNA into protein. Yes, here is the ribosome. It attaches to the RNA, and from the RNA it reads and it converts that code into the protein by adding amino acids one by one, like here. If you can see, from the DNA, with the help of RNA polymerase, we are getting, from the DNA by the help of RNA polymerase, we are getting RNA. We call it mRNA or messenger RNA. mRNA carries the code of a particular protein. We call it transcription. And this mRNA is bound by a ribosome, and ribosome starts reading the mRNA 3 nucleotides at a time. Three nucleotides together, we call it codon. So it reads 3 and adds 1 amino acid. Amino acid is carried by a tRNA. So ribosome reads 3, adds 1, 3, 1, like that. A chain of amino acids is made. This chain we call it polypeptide chain, and this chain, when it folds together, it forms a protein like insulin, keratin, hemoglobin. Yes. The decoding of mRNA into protein, we call it translation. So in central dogma, we see two processes, from DNA to mRNA, from mRNA to protein. We call it transcription and translation. There's another concept, because a DNA which exists in the cell, it just not stays like that. It gets passed on to the further cells. Yes. For that purpose, DNA gets replicated. So in the central dogma, we have DNA being replicated whenever necessary, DNA being transcripted, and RNA being translated. We call this theory of central dogma, where the genetic information is transferred, is flowed into the protein. Here, let us watch a video and understand this concept. A journey from DNA to protein. Here is a cell, and inside cell we have nucleus at the core, and inside nucleus we have genetic information in the form of DNA. DNA, when it compiles or condenses together, it is called chromosome. Yes. If we open it, we can see the DNA. DNA is condensed by the help of histone or protein. Yes. In DNA, we have multiple genes. Every gene codes for a particular protein, and for protein to be synthesized, we need first DNA to be transcripted to RNA. This is the translation process. Yes. Polymerase is synthesizing a new strand of mRNA by adding nucleotides A, T, AUGC. This is mRNA made of AUGC. It carries the code from cell, from the nucleus. Yes. Unnecessary parts of mRNA is removed sometimes, especially in eukaryotes. After it is synthesized, it travels out from the nucleus into the cytoplasm, where ribosomes are present. Ribosome comes and attaches smaller subunit first, larger subunit later, and then comes the tRNA, which carries an amino acid. The tRNA has anticodon sites. These are the anticodon sites. You see it? Yes. It comes and reads 3 at time, adds 1 amino acid. You see these 3? Yes. It reads 3 codons, add 1 amino acid, and then forms a polypeptide chain, a long chain of amino acids, like beads on string. Yes. All these beads, when the mRNA is completely red, it stops making polypeptide. It folds and becomes a protein. That's how all the proteins in our body are made. I hope you enjoyed this session. Thank you.